Hi, welcome back to the Between the Vines. I'm Jennifer Phillips Russo, the Cornell Viticulture Extension Specialist and Team Lead of the Lake Erie Regional Grape Program. Our program is made up of specialists from both Penn State University and Cornell University. This is brought to you and housed at Penn State, but it is a Cornell Extension team. And it is our job to bring research-based information to our grower-based knowledge to help with productivity and sustainability of the grape industry in Western New York and Western Pennsylvania. So today we're going to have a podcast that addresses some of the questions that you have submitted in regards to the drought-like conditions we experienced this last growing season. We have with us two very special guests, Dr. Terry Bates from the Cornell Lake Erie Research and Extension Laboratory. He is our director and has been basically co-host of this podcast for the last 5 years with us. And we have Dr. Alan Lakso, who is emeritus faculty at Cornell University. Thank you for joining us, gentlemen.
Thank you.
Thanks, Jen. Nice to be here again.
Again, thank you, Terry's. Alan, I could read what you have online, or I could ask you to talk about your career that you have provided to our industry. Which one would you prefer?
Well, I can keep it probably shorter than the stuff written online. So. Yeah, I did my PhD out at University of California, Davis in the Vitinology department, working on grape acid metabolism with temperature effects. So been interested in climate and effects on grapes for a long time. And I came in to Cornell to take a faculty position in 1973 and been there ever since. Retired 11 years ago, but still interested in grapes and how they respond to the environment, and still doing a little research and writing and giving a few lectures. So happy to be here, and like to talk about grapes and weather. So
fantastic. It fits right in. And that is a 52-year career. That's amazing. Thank you so much for your contributions that you've given to our industry over the years. Appreciate that. So
I like to just say that I'm really happy that Alan's on.
Yes.
When I started at Cornell, I mean, Alan was my grape mentor, really. I came from Penn State in the plant nutrition lab and did not work in grapes. And when I got to Cornell, I mean, it was Alan that taught me basically everything, all the basics of grapevine physiology that we've been able to use in production management since then. So I'm really happy that Alan's on, and I was there in Fredonia when they did all this really good work on Concord and Niagara water relations and cover cropping and all sorts of great physiology stuff. So
fantastic.
Really
Are you blushing yet, Alan?
Well, there's a fantastic group out in Lake Erie, both Fredonia and Portland now, and it was really a pleasure, and it made the research so much better with the support that we had there for so many years. Just the types of, we carried out a study for 12 years on pruning and irrigation interactions that very, very detailed and just could not be done anywhere else. So I have a great appreciation for the group out there.
Thank you. And we appreciate you joining us today, even in your retirement. So you are the expert on water relations with our grapevines. And some of the questions we've had was we definitely experienced some drought-like conditions. And Terry can attest, and I can even let him talk to you about what he discovered in the railroad block during the harvest. Do you want to start with that, Terry, to give like a little bit of a background of where we want to take this?
Yeah. So we had a very dry summer out here. The spring started off fairly wet. So everything when we started to, look at berry weights and crop estimation, we were on track for where our predictions were. And then we had a very dry summer, berry weight was lower, and that lowered our crop prediction. And what you're talking about in railroad block, when we harvested our 6-acre railroad block, we did it in 2 different days. And it had rained, I think, 3/4 of an inch between those 2 harvests loads, and you get when we do our spatial mapping of yield, you just see this big blue streak right in the middle where the second load came through after we had enough rainfall. I think the biggest question we're getting from our growers are, we had a dry summer, so sugar was high, berry weight was low, and what can they expect for next year? We're already getting reports of very uneven paraderm formation. You go out there, it looks like the vine is really good and has lots of brown paraderm, but then you go to prune it and it's half dead.
Paper.
So just kind of like, what should we expect in terms of vine size response and fruitfulness response of the buds that we leave? I think is the biggest question.
Yeah, this was a very unusual drought year. And normally we start out with a wet spring and things go pretty well. One of the real keys about drought effects on grapes or many of the other fruits as well is that the strongest effects of drought tend to happen when you're getting the drought during the early season, during the early shoot growth and early berry growth and berry set. Um, and that's because that type of growth is by cell division. And cell division is more sensitive to drought stress and needs a better water status than later on, where after the shoots have stopped growing, then you're just maintaining them. Or with the grapes, trying to produce new cells is more sensitive than just accumulating sugar, say after raisin. So this year, and I'm not sure exactly when it really got going for you here in Geneva. It was really quite wet until right about the middle of June, maybe 10th to 15th of June. So most of those early season processes were already done, or there was enough soil moisture to maintain those early season processes. Um, and a lot of pruning weights and things like that would be determined fairly early. Um, and so I wouldn't necessarily expect, if under normal conditions or with kind of average soil, I don't think that it would have gotten that dry that quickly. But now, of course, you've got the gravels, and they can dry out fairly quickly. Only maybe 18 inches of real water holding capacity in a lot of cases. So, of course, then you've got heavier soils too. So you obviously would expect the lighter soil, the gravels, to dry out more quickly. So I went back and I looked at we did a study that we compared minimal versus balanced pruning over many years, and with and without supplemental irrigation. And I looked back, 1991 was a year where we had a real significant drought, um, more like in the lines of 2016 and this year. And I looked to see what was going on with not only the pruning weights and response to irrigation, and there were several factors that we found. One was that overall we did not see a really dramatic effect on the balanced pruned. We also had an 80 node standard pruning. We didn't really see much an effect of the irrigation or drought in that year or over the whole 10 years on the normally pruned type of vine. The minimally pruned vine did respond significantly to the irrigation or drought stress. And so it turned out we had, at the time, Barry Schaefer was the economist working with us, and he did an analysis, and we came up with the conclusion that irrigation would be economically viable or would be worthwhile economically, but only for the minimally pruned. And I think part of this was probably a combination of two things. With minimal pruning, you have so many nodes that you get a tremendous number of shoots, and so they all pop out, and your canopy develops very quickly, and it captures a lot of sunlight, which allows the minimal prune vine to produce a lot. But when you're capturing a lot of sunlight, it's also using a lot of water. And so we found in that year that the minimal prune that weren't irrigated tended to get into drought stress earlier. The normal pruning, you have fewer shoots, they grow longer, so it takes longer to get to a full canopy, and they don't use as much water early in the season. So we were finding that both the minimal pruning used more water, but also they had heavier crops. And so in this, we didn't do any crop adjustment on the minimal pruned ones. And so we found overall that there, in terms of yield and pruning weights, the irrigation had very, very minimal, if any, effects on the normal pruning approach. But with the minimal pruning, we did see that it turned out about a ton per acre every year. We had an additional with the supplemental irrigation. So it was about one ton per year. And I believe the conclusion was it was just enough to pay for the irrigation system, but probably not that much more. Just avoiding the loss rather than making extra money with it.
Question about that? Is it
I'm
all right
sorry?
if I ask a question about the minimal pruning that you did with the irrigation? Did you have a trial that didn't irrigate the minimal pruning? And what was the response of the vine if you did?
Yes, we had both irrigated and non-irrigated in each of the pruning systems. And yeah, the generally we didn't see, well, we got some trimmings, so we don't really have pruning weights on the minimal pruned. So those comparisons were minimal. You couldn't really tell anything from that. But in that year 1991 that we were talking about, that we did get by the end of the season, we were seeing basal leaf senescence and leaf drop in some of the most severe spots in the vineyard with the minimal pruned. With no irrigation, we were actually seeing some fruit shelling off by harvest. So it was pretty severe in some spots. The interesting thing was that one of the conclusions with the minimal pruning was that overall, yes, it was about a ton per acre, but we had four different blocks within the overall vineyard there. And one of the blocks showed no response whatsoever to irrigation. It just produced beautifully, high yields and good sugar, and everything was fine whether or not they had irrigation. Um, but another block, not that far away from it, actually showed the strongest effect. So the overall effect of the irrigation really was to make it more uniform. So if we only looked at that one block in the driest spot, we would see probably a more closer to a 2 ton per acre, uh, advantage with the irrigation. This is supplemental irrigation. It's not always used. Um, so one of the things that we've seen across quite a few different trials that we've looked at it in grapes and vinifera too is that irrigation can help provide more uniformity. And that's one thing in New York. We've got such a range of soils, and even within a vineyard, there may be three different soils, and then they might even vary a bit within the type of soil. And so we have a lot of soil variation, which means if you're going to have a drought and the water holding capacity varies that much, you're going to see a lot of varying effects. So one of the things about irrigation is that it helps make things more uniform, uh, and so that was one of the major uh, things that we've found from that, and we've seen in other cases too. So, sure if we're getting off topic.
Yeah, no. So, okay, so based on the results of that study, plus growers often will cite about the expense of irrigation and lack of good water source for their vineyard. So most of the management in our area is water conservation through floor management, and I know you've done a lot of work on different floor covers and cover crop width. You want to talk about that a little bit?
Um, Yeah, actually, before we get away from that, I did dig out some of this older data from the 90s on that trial, and um, I did find the paraderm counts from the vines, and the balanced prune vines, regardless of whether they were irrigated or not, all had over 500 nodes of paraderm on the vines, and so it didn't. Again, it didn't seem to have a significant effect of the irrigation or drought. Um, in the case of the minimal prune, though, we did see a uh, quite a significant drop off, and we found some down in the neighborhood of say 225 nodes of paraderm, and some up more around 450 nodes after that drought in 91, and it did correlate quite nicely in kind of a fairly linear manner with the number of shoots per vine in 92, the following year, or and also the number of clusters per vine the following year, and then because of that, the yield per vine the following year. Um, so there was a excuse me, a clear effect. The irrigated ones were all over 400 nodes of paraderm, while the non-irrigated ones, as I mentioned, there was some variability between the blocks. They varied from about 350 down to about 225 nodes, with a consequent effect the next year on shoots and clusters and yield. So um,
So what would you say the ballpark like Ravaz index? I know you can't calculate the Ravaz index because you don't have pruning weights on minimal prune, but like ballparking it, uh, were they overcropped, balanced, undercropped?
um, well, they had crops that were about the same as and the over the 10 years, it was striking to me. I thought because there wasn't the pruning and the balancing, that the crops would be going up and down and up and down a lot more with the minimal pruning, but we found less variation in year to year crop levels in the minimal pruning than we did with the balanced or 80 node pruning. Um, and that was quite striking to me. And we could have a physiological discussion about why that is. I'm not sure. I'd be speculating, but that's what we did find. So we didn't see the really major drop-offs. And I think part of that is even with the drier years, um, so much of we get that early canopy development, and so when the berries are going to be set right after bloom, the canopy is really full, and the shoots many of them have stopped growing, and probably a few are still growing, but the shoots really aren't requiring anything. So there's a lot of carbohydrate that can go directly to setting berries. So I think maybe that's why there's um, that consistency of cropping that we saw. Now that may not be the case, and I'd like to ask Terry whether or not you see that in across the commercial vineyards.
Yeah, I don't know if anyone who does minimal pruning anymore. Um, Most people have dropped back to some kind of mechanical pruning, hand follow up, somewhere around 120 to 150 nodes.
Yeah.
So,
Well, and I had a grower ask me the question, and you sort of touched on this, but you said that you also looked at cluster counts on the non-irrigated vines that you had in that experience. Did you have lower cluster numbers on the ones that didn't receive as much water as the irrigated? So they're worried that this season they're going to have canes that only have 1 to 2 clusters instead of 2 to 3 clusters on them. So they're trying to figure out in their pruning techniques if they should leave up more based on what the fruit that we can guess that the vine's going to do next season.
Um, well, yeah, as I mentioned, we have treatments, but it sounds like in many cases the industry is in between them.
yeah.
So
Mhm.
uh, we have to try to guess with the uh, with the minimal pruning because there was a big range in the number of paraderm counts. Um, then there was a fairly big range in the number of shoots per vine the following year and the number of clusters per vine the next year. There didn't seem to be quite as much change in the number of clusters per shoot, um, but it seemed to be more the total number of shoots that came out that seemed to have the biggest effect. Um, so yeah, it's a little bit hard to say. I would be concerned. There was not on Conkers, but on some other um, wine grapes in Europe. They did a study and they were looking at the water stress and they measured the actual stress in the vines and they were looking at it in the period right around bloom and shortly after bloom and um, they found a very quite strong effect of that water stress um, in in your in that given year with the weight of the berries and the pruning weight uh, related to that just that period shortly after bloom, and then in the following year the bud fertility or um, clusters per bud also went down quite clearly with more water stress in the post bloom period of last year. So if uh, if the water stress did develop in that post bloom period early enough, we would expect some effects on the bud fertility. And then they also looked at the berry number per cluster in the following year, and that also even went down about 25% with some stress. So um, there is evidence not only in Concord but other in wine grapes that if you get stress in that fairly early post bloom period, that it can affect the bud fertility and berries per cluster the next year. Of course, there's could be some complicating factors too. If there's some stress and it slows down the shoot growth and the leaf development, um, the exposure of the buds during that season is also that's an important period. So if you don't have as much vegetative growth, you may have better exposure of the buds, which could counterbalance some of that stress effect. So these are very highly integrated plants, and they respond to not just one or two of the factors that we might be focusing on. Um, so sometimes you get kind of surprising results, and when you start looking at not only the water, but also what does that do to nutrient uptake, uh, and what does it do to the canopy microclimate, those sorts of things. So it's a little bit we can speculate, but we have to keep a big grain of salt handy.
Well, you bring up a good point because another grower submitted a question stating that their crop estimation for a certain block was about 10 to 11 tons per acre, but it only came to about 7 to 8 tons per acre because of the drought, the effects of the drought, excuse me. However, they're curious if the grapevine before the drought occurred took up enough nutrients out of the soil to sustain the 10 to 11 ton. They're actually asking me about what should their nutritional amendments be this coming season. Should they consider it a 10 to 11 ton draw or a 7 to 8 ton draw?
Uh, well, I think you're asking the wrong person about mineral nutrition. That's been the
Dr.
one.
Bates?
So the one part of physiology that I've struggled with a lot, and I'm very not really willing to to make any suggestions that anybody might take seriously. So I'll turn that one over to Terry.
We have great growers, don't we?
You know
They come
that
up with great questions.
yeah, I think the question is so growers talk about like how what were the nutrients removed and because they're under like a replacement management. Like if I can calculate the number of pounds of potassium that came out, can I just replace that in the vineyard? I think the drought has an additional effect of if I have dry weather and I'm slowing down canopy photosynthesis and I'm slowing down, say, root growth, now I'm actually inhibiting my ability to take up those nutrients. So you have your removal effect. Yeah, I guess I would argue that the grower had they predicted a 10 ton per acre crop, got an 8 ton per acre crop, and that's really all water. It was just that the berry weight was lower. So the nutrients that went into that fruit is equivalent to what would have been a 10 ton per acre. But I think that the other factor is, do I need to put on a little bit more fertilizer because my root growth was restricted and I'm kind of trying to make up for that for the next season. So I would put on a little more.
Yeah, that sounds logical that if it is a late season effect, a lot of times, these things have been taken up into the fruit, and then the water just wasn't there at the end to blow up the berry. So in a lot of cases, I think in 91 we calculated once we hit 1st of September that the bricks continued to go up, but that was because the berry size was going down. When we calculated the amount of sugar per berry, no additional sugar went into the berry in the last 3 weeks. But it was the berry size went down so that the bricks concentration went up.
Yeah.
So that can be a little bit deceiving when we just look at the bricks. So it would have been nice, I guess, if somehow could have gotten the actual amount of nutrients in the crop, because it may have been more normal. We've seen that with some stress studies also in apples where everything seemed to be fine, and that at the end they didn't expand as much because they didn't have as much water, but some of the mineral nutrients weren't affected very much. We did get mineral nutrient data on that study, but I haven't been able to dig it out quite yet since you asked about it. But I will need to go look at that, uh, see if I can dig that out. But my recollection was that it didn't seem to have dramatic effects on the petiole analyses. Um, the 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 effects were primarily due to minimal pruning versus balanced pruning, not the irrigation. So
So there's a quote that goes around our industry every time we have a dry year about early but incomplete paraderm formation. And I don't know if that came from you or Bob Poole, or do you recollect
that
that
sounds
saying?
that sounds like something Bob would say. Yeah.
Because I think we're seeing that this year. We see a lot of variation, which I think you've already touched on in your trial, saying one block the irrigation had no effect, and the other one it did. And I think with our variable soils, we're seeing that variation in paraderm formation. But then again, also we're getting reports now with growers that now out pruning, they're starting to cut into the canes, and they're realizing that they're dry, and don't seem viable, where they look fine on the outside.
Yeah. I guess I mean it seems sort of logical when you've got a like this year was very sunny, warm, dry. You're not getting a lot of vegetative growth, in the mid and late summer. So once the shoots have stopped, then I think that general initiation of paraderm seems to occur earlier when when you have a wet or a dry year, especially a dry, sunny year. So the idea that it got started early seems, very reasonable. And then when we ran out of water, then they weren't able to continue that development because the paraderm is an active anatomical tissue. So uh, it takes water and support to actually grow it. It's not just drying out. So that observation, that makes some sense. Um, we did get samples in the fall to look at cane starch levels, and I was looking at that earlier, and um, couldn't really make much sense out of it from the with the balance pruning where there wasn't really any clear effect, but there was a lot of variation. The I don't remember the units, but the values were from like 3 to 20 or something like that, and
Mm.
each block was different. So I'm probably a sampling issue or something like that. However, with the minimal pruning, all of those, whether they're irrigated or not, were very low. They were something like two or three. I don't remember the unit. Um, so it was about like, the balance were like 5 times higher concentration of starch. But I suspect that might be due to the fact that the size of the shoots on the minimal pruning are really quite short, and they grow and then they stop, and they may just not have the ability to accumulate that much starch. Um, so we did have the look at the low temperature exotherms, um, and we had help doing those. And I pulled out the data on that, and after 91, um, we saw, well, a couple of effects. One was that the balanced pruning, again, irrigation had no effect. At the time we did it, which I think was in probably about this time of year, maybe a little bit earlier, the LTEs were about minus 12, minus 13 Fahrenheit. Um, when with the minimal pruning, and the strongest effect was all the minimal prune ones had less uh, um, or less negative LTE. So they were down more like about 8 and 9 or 7 and 9, and uh, the minimal pruning with the irrigation was pretty much the same as the minimal pruning without irrigation. So the major effect was that the minimal pruned uh, buds were slightly less hardy than the others, but the irrigation didn't. We were a bit surprised, but the irrigation didn't seem to affect it. Again, this may have been, the more basal buds developed earlier, and so perhaps the development occurred before the water stress got too severe.
The same. That was about a 4 to 5 degree difference too, yeah?
Yeah.
7 and 12, yeah.
So,
Terry, you're making notes.
Yeah. I always take notes with Alan when Alan is around.
Did you want to go on to your question, Terry, about root distribution and fine root growth and death with different cover cropping or floor management strategies?
Sure. I mean, that's a big ball of wax
It is.
between
Or
the
maybe
different
that's another
trials that were
podcast.
done. Yeah, right. Just all the floor management studies that um, you did. Yeah. So I was thinking along the lines of you did a row middle management study. You actually did two of them with Bob Poole. Then you had that cover crop width trial.
right.
And then that was followed up by all the root biology stuff with Dave Eisenstadt and the mini Rhizotron tubes, and that's a lot of information.
Yeah. Yeah. Let's see. Um, let's go back, yeah, to the 1980s. I don't know if that's a different geological era or not, but uh, the actually, I was if you want to feel old, what you need to do is look at the record as to what the CO2 concentration of the atmosphere was when you it was it was
When you
300
started?
it was 325 parts per million when I came to Cornell. So uh, that gives you some idea how long I've been around. Um, but in any case, yeah, we did a study at Rich Ertles out at Silver Creek, and it was row middle, and we had different. I think there was a single roundup, double roundup, one before and one after bloom, and there were several different ones. The study gave some kind of confusing results, um, but we began to sort it out when we were taking soil moisture measurements. And what we found was that we had different blocks that had been laid out, uh, and it was going down a slope. So we thought fairly logically that it'd probably be drier at the top, and so we blocked it. So we had some blocks at the bottom and some further up the hill. But it turned out the variation in the soil and the water availability went kind of diagonally across the slope. So the treatments, for example, the single Roundup gave better results and better growth and yields than the Roundup than the double Roundup, which was more conservative in terms of cutting back on the any competition. But it turned out that the way the experiment was laid out was that the double Roundup had more plots in the dry areas, and so when we expressed it just on a soil moisture basis, everything fell in line. So we followed that up with, and Bob Poole led the study, and I worked with him on the looking at growth and the water status and that sort of issue. And so we did the next study in the 90s at the Fredonia lab, and Um, we found that we had 10 different uh, treatments. We had mulched, straw mulch, we had cultivated, we had Roundup, post bloom Roundup, which was what the plot had been before. So that was kind of a continuation of the standard. And then we had different types cover crops. We had orchard grass, which is pretty competitive, and we had it by without doing anything, and then also mowing to see if mowing would have an effect. Um, and we had clover and vetch and annual ryegrass, and then annual ryegrass that was killed with herbicide. And I'm probably forgetting one or two in there. These are row middle with a clean strip under the vines, and we followed that for several years. And um, a couple of the main conclusions out of that was um, that um, the most competitive were like the orchard grass that was the strongest competitor, and uh, but what we found was like on pruning weight, the very first once it got established, that first year with the established cover crops, um, showed quite a strong effect on reducing um, the pruning weight that from say the Roundup or the mulch or the trashy cultivation. Um, but we found over 2 or 3 following years that the pruning weights came back up. And after about 3 years or so, I don't remember if they got exactly back up to the same level, but they got up, pretty close to what they had been before. And so we did some trenching to look at the where the roots were. And when we looked at the area that we would call the zone of competition, so it would be just out from the herbicide strip to the row center and down about maybe 18 inches or so, where if you had grass, that's where the grass roots would be and the vine roots. Um, and we found that in the case of the Roundup treatment or see, I think we did mulch also. We found that about a third of the grape roots that we found were uh, in that zone of competition. Well, in that zone. Of course, if we didn't have competition, we had about a third of the vine roots were in there. Uh, However, when we had something like the orchard grass, we found that there were only about 10% of after 3 years, about 10% of the grape roots were still growing in that area in competition with the grass. And almost all the other roots had grown deep, either directly under the vine or actually deeper and underneath the cover crop, but be lower than the cover crop roots. So the vine basically couldn't compete with the cover crop roots, and so the roots would grow somewhere else where they could. And in these deep soils, they were able to go deeper, both right under the vine and under the alleyway. Um, and so, uh, the vine roots. I've seen this in other vineyards where the vine roots will go wherever the conditions are the best. And the uh, one thing about vines and a lot of our fruit crops, they don't have a lot of, they don't have huge. They have big root systems and they can go a long ways, but they're not very dense. And so one of the thing that we looked at is what's called the root length density. So, what length of roots do you have in a volume of soil? And you can compare different. It turns out if you're comparing like a grass like fescue or something like that to grapes, the fescue will have 50 to 100 times more roots in that volume of soil than the grape does. And so you can imagine that that one grape root is trying to compete for water with 50 or 100 other grass roots around it. So you can see where the grapes can't compete very well with the grass. Um, but since a grape is doesn't have inherent structure, the top doesn't grow like a tree. It goes over whatever is there, and the roots are about the same. They're called opportunistic, so they just go wherever the best conditions are. And so in that case, we found that of course when you first put the cover crop in, it looked really bad, but then since the soil allowed the roots to grow deeper and to avoid the competition, then the vines recovered fairly well and sort of came to a new equilibrium with that cover cropping system. But the initial results, looked like it was, should not put should not put that orchard grass in there. Um, we we did one other question was is there's a competition for water or nutrients or when and I was looking at the water part and we decided to do a little study where we took some pots. They're fairly deep pots. They're used like by the flower industry. They're maybe about 8 inches or 9 inches in diameter, but they're about 15 inches deep. So when we put in the trials, we dug out the soil in a hole and put a double pot into each of the plots and filled it with the same soil. And then when all the things were planted, we had the same grasses or whatever planted in that pot. And after a good rain, so everything was wet, we could pull the pot out, weigh it, and then put it back in place. And then a few days later, 2 or 3 days later, we'd pull it out and weigh it again to see how much water that plug of the grass or whatever the cover was had actually used. Um, because there's been studies where people said they measured the water use rates from different kinds of plants, and they said, oh, some of them use more water than others. It's not surprising, but the when the plants are actually in the vineyard, if you think about the cover crop in relationship to the vineyard is a huge windbreak uh, for the cover crop. So the cover crop sees a giant windbreak every 9 feet. So the wind is really, really low in that area. And there's some microclimatology things that suggest um, that the water use of the plants are primarily going to get driven just by the amount of sunlight that they get. And so we measured, and so we basically found that any of the cover crops that were green active uh, used about the same amount of water. And uh, if we uh, uh, if we did something like mowing apparently doesn't seem to help if, like say with your lawn, it's green, a little long, you mow it, it's still green. It's going to use the same amount of water. Um, if, however, the grass or if it's a cover crop, if you mow it fairly low and afterwards you look at it and it's kind of yellow or, pale green, old leaves and things like that, that will reduce the water use. But after about a week or so, you'll get more regrowth, and then it seems like the water use goes back to about the same. So the mowing is, uh, in a lot of cases, if it looks green after mowing, you're going to get maybe a temporary effect, but it's not going to be a major reduction in water use. Knocking it back when things either went naturally dormant or if we knocked them back with herbicide, then they would definitely use less water. Um, but it had to be something, that was not just a temporary thing like mowing. Um, so yeah, so we learned a few things there, and then we tried the
So if you put in a cover crop, you need to be patient to try to get those roots to redistribute in the vineyard. And then, do you think once you have those deeper grape roots, does that make the vine more resilient to drought?
um, I think it depends. It depends a lot on the soil structure you've got. Um, because if you're in a soil that say is only about 18 inches deep, or has some restrictive layer where the roots can't go down, then if they can't go down, and then box them in on the sides with the cover crop, then you put them in a smaller box, and you can limitation and root development. Um, in the gravel, there, once you get below about 18 inches, at least some of the spots that we were digging, that there wasn't a lot of water or nutrients down uh,
Mm-hmm.
in that very deep spot, or at least say, the next 18 inches or so. It could be some. So it depends on what that soil structure is. If it's, if you've got water, like 3 feet down or 4 feet down, and you drive some roots down there and they find the water, then, maybe it may be even better off than what it
Mm-hmm.
was. So, um, yeah, the soil structure and the grape root behavior was an interesting observation where we looked at Niagara. We had Niagaras and Concords growing next to each other, and we did root trench in the alleyways between them and looked, and we had found that it was a trial comparing Concord and Niagara's on. We wanted to establish, looking at Niagara establishment and whether irrigation was helpful in that case. And um, what we found when we dug down was, first of all, the Concords did not respond. These were normally pruned Concords, did not respond to the irrigation, but the Niagaras, even though they were fairly well established, did respond nicely to the irrigation. And when we dug down, we found that about two-thirds of the Concord roots that we could find were in the top 18 inches, and there was more organic matter in that upper layer. Um, but uh, the Niagaras were just the opposite. It seemed like maybe a third of the roots were in the top, and two-thirds of the roots were deep. But at least in that section, those, being deep didn't necessarily give them much water,
Mm.
and so they they apparently naturally grow deeper, at least from that one observation. And when they did, they got into drier gravel, and so they actually responded to irrigation. Where the Concords that had just a completely opposite root distribution did not seem to respond because presumably they had more roots in the where there's more organic matter and more water holding capacity.
Mm-hmm.
Um, but the other outcome from that Niagara study was that um, when we had irrigation, um, the in the let's see, I think it was the second year, uh, we had really really good strong pruning weight. The very first year, that I guess it would be the second year of the planting. Uh, and Rick Dunst, our colleague at the time, said, um, "Well, we're supposed to do balanced pruning on these, but this pruning weight says we're going to have 10 tons per acre in the third year. And do you really want to do that to the vines? Maybe we need to find out. I don't want to kill the vines, but at the same time, maybe we need to find out whether or not they can actually, handle that. If it had that much growth, it should be able to handle the crop. So we went ahead and balance pruned, and with irrigation, they had 10 tons per acre in that first real crop, and uh, and then we thought, oh, it's going to the pruning weight will crash, but the pruning weight came back, like 2.5 pounds or
Mhm.
whatever it was. It was good pruning weight. I think we got 8 or 9 tons that very next year, and so for the first 4 or 5 years, we were 9 or 10 tons per acre, with the irrigation. Without the irrigation, we had the big up and down. And if we had maybe 4 tons, and then the next year went up to 8, and then it crashed back down to 4. It couldn't, without the irrigation, it couldn't both grow the vine and the crop.
Mhm.
And, um,
Make sure you tell Ted Taft that, Terry. We keep wanting to put some irrigation into some of our Concord plants.
okay, well, now I don't want to get in a fight with. Okay, yeah. Let's see, the other study was the cover crop width, where we
Yep.
had essentially no cover crop to complete cover crop lines too. And um, as you'd expect with the pruning weights were kind of linearly related to how much bare soil there was. Um, and we did we had the hypothesis based on the other stuff was that if we had different levels of cover crop cover, that if we irrigated, we could counterbalance, the competitive effect. The interesting thing was, and I don't know that we ever really got a firm conclusion as to why, but it seemed like that seemed to be the case within the alleyway, the range of cover within the alleyway. But once we got under the vine, even with irrigation, the under the vine um, of cover actually had a negative effect on the vines. And my recollection, again, dig it out, but my recollection was that we looked at the nutrients because that's the obvious other factor that would, and it wasn't clear that it was an obvious nutrient competition effect. So I think that one kind of was left up in the air as to, I guess the conclusion was it's detrimental to go completely under the vine, but the reason for that wasn't clear. Uh,
Right. I think what I remember is even if you had just a 1 foot or a 2 foot weed free band underneath the vines, the irrigation then could be beneficial.
yeah,
And that would make sense based on what you were talking about earlier, where you found 2/3 of the roots in that weed-free zone, like if they had just
yeah,
changed their distribution.
yeah. So, in any case, I think those are the major conclusions from the
Yeah.
studies that I can remember. I have to go back and really look through all the data to be sure. And if if there's specific questions that I might be able to answer, let me know, and I'd be happy to try to dig through the data. We've got accumulated a lot of data, and um, both our stuff, and then also the crew at the lab generated unbelievable amounts of data with all like the paraderm counts, and I reported at meetings uh, this type of data, and our colleagues are saying, "How did you ever get that kind of data?" We're just damn lucky to have a crew like you guys have. So, very appreciative.
Yeah.
Very appreciative of all the work that you've done that has informed management in our region for the last 52 years. That's amazing to me. Terry, did you have any other questions? Because I don't want to take all of Dr. Lakso's day. So
Uh, the the one last thing, if there is any like broad conclusions from the Paula did all that work with the mini rhizotron tubes, finding out when fine roots grow and then die and grow and die. And I mean, I know there was a lot of data there, and it was year to year variations. Is there any big conclusions that ever came out of that work?
Yeah. um, for those that are not too familiar, we worked with Dave Eisenstadt from Penn State, who is a root specialist. And so we did a study where we came in and worked within the minimal versus balanced pruning study. And we used a technique called the mini rhizotron, which you may not be familiar with, was using it was a clear plastic, about a 2-inch diameter tube, like a PVC tube, and we'd put maybe three-foot tube, and it's plugged on the end, and we stick it down in the ground under the vine, and the roots will grow around that tube just like it's a rock, uh, and because that tube is clear, we would slide a special camera down the tube that would take a picture. You could see a video picture where you could see out through the tube and see the roots growing around it. So we could go back and drop it at different points as you go down the tube, and so we knew at different depths when the roots were actually growing, and then by looking at the pictures of them, we could see, they start growing, they're white, and you know, physiologically, they're really active, take up a lot of nutrients and things. And then when they start getting brown, they lose some of their function, and then eventually they'll die and disappear. And so we could keep track of when do they really grow, and how long they live, and then are there certain things like maybe the phenology of the vine, anything happen special at bloom or veraison or after harvest, and those sorts of questions. Um, and so Dave's group worked there, and then we worked with them by having the trial where we're keeping track of all the growth and development of the vine, and we're monitoring the stress and the physiology of the vines, um, and so it was very interesting to see what the going on with the roots. And of course, we went into it saying, okay, we you know we should be able to see a relationship between, maybe when the shoots stop growing, this is when the roots can grow. Uh, and, we know the pattern of root canopy development and the pattern of fruit development is quite consistent. It may, stop a year earlier in a dry year, but, it's a pretty consistent pattern. And so we expected that the patterns of when roots grow would be, quite consistent as well. It turned out the roots will grow whenever they damn well please, it seems. Every single year had a different pattern of root growth. And we could not necessarily relate it to, it didn't correlate very well with, say, the growth of the top. Um, it did not seem to relate too well to whether it was, say, a sunny year or a cloudy year. Um, it these are the new roots. They're the most active ones. Um, it was actually surprising, quite disturbing, since we wanted to understand what was going on, that these patterns of root growth were way more uh, variable and did not seem to correlate with what was going on the top of the plant. And we couldn't necessarily, we thought, well, we could be able to see something about the physiology of the top. It seemed like the maybe the conditions of the top, like the crop level. If it was a little lower crop level, you got more root growth, but it didn't necessarily make it, more consistent. It was variable every year, and so we did a lot of work on that, and it was actually quite surprising how little we could explain uh, in terms of the behavior of the top of the plant by what was going on in the roots. Um, we did, of course, find that when it was drier, we produced fewer roots if it got really quite dry. Um, although there's been studies that it's kind of variable in other vines and fruit studies. Um, there are ones that have shown, early season root growth, even before bud break or around bud break, and then very little growth in the middle of the season, and then a big flush of growth after harvest in the fall. We did not really see that post harvest. We didn't see the early spring uh, flush of root growth, but that seemed to be primarily because we've got cold spring, and we did see one little blip of early root growth when there was about a week of like 70, 75, close to 80-degree temperatures in one year. It was a very unusual warm period, and we saw a little bit of root growth, a little flush of root growth, suggesting they probably could grow then, but that was, if the temperature was warmer, consistently. Um, but we mostly saw the root growth occurring between bloom and veraison. Um, and after veraison, especially if you had a good crop, we didn't really see much new root growth occurring. Of course, you had the roots that had grown just previously. Um, and again, we did not really see much in the way of post-harvest flush of roots. And I think maybe that related again to the climates where in some cases, people were harvesting, they'd harvest the vines and there'd be another couple months of nice warm weather. And so they might get, it might be the vine canopies can produce enough carbohydrates and support more root growth. But in the Concords and here we did not see that. Yeah, so I think it's maybe a combination of the climate and timing of when we get harvests. We're fairly late in the season, and maybe also they're natives to this area, and perhaps they're shutting down at that time of year rather than trying to grow more roots. But even the total amount of roots varied amount from year to year, and so um, it was it was interesting, but kind of confusing as to what in terms of any management. Just not clear what it was going to tell us that would help us to manage it much better. So yeah, that was that was one of the more confusing and interesting things that we did.
Wow,
Uh,
I can imagine tracking that growth and counting. That's a lot of work.
yes, and that was a nice thing that we let Penn State do for us.
Yeah.
They did a great job.
Louise Comas, I think she was the one that drew the straw on that one.
Yeah,
Yeah.
she did a lot, and
Well, great.
So the drought-like conditions from this past season shouldn't have as big as effect as some people are thinking, dependent on the soil type that you have, based on that it happened after bloom and fruit set, really. Is that just about sum it up?
that's yeah, that would be my guess on that. Um, Again, I'm not really sure exactly when it really started developing, and in a lot of cases, because the roots, conquered roots and grape roots or rootstock 3309, they can go really deep, and so there's so much variation as to you might have the same soil, and some vines will be able to get some roots down, and other vines don't push roots down there. That um, I would expect it to really show the variability in the soils. Um, and when you have a average or more wet season, I would think there would be probably less variability um, because all of them would have adequate water.
Mhm.
I know
Yeah,
here
anecdotally, oh, I'm sorry, go ahead.
in the Finger Lakes, as I drive around and look, we were really dry, but there were some spots with quite large canopies, and everything looked great, and others where there were some vines that were quite small, initial defoliation, and so they were really reflecting the soil variations a lot in this case.
That's what I was hearing from growers. If they were on a heavier clay soil, that they fared a bit better than those who are on the gravel soils during this season. Not by much, but a little bit better.
Yeah, I think that yeah, with the clay type soil, sometimes see it will do well, and they'll put a little big vine, and then when it runs out of water, you've got a big vine using a lot of water, and then it just like the valve shuts off, and they can have some pretty dramatic collapse in vines. We'll see that sometimes where within the maybe the not just a spot, but broader when you have a wetter season and you can grow big vines because of the season's wet, and then when it gets dry and hot and it shuts off, then you've got a vine that not only isn't getting that much water, but needs a lot of water because it's so big. And so they're doing great and they collapse. So it's difficult to make consistent conclusions when you have extremely variable soils, and then you're imposing extremely variable climate on top of it, and trying to manage it. So it's a real challenge, and I really respect the growers that can figure out how to handle that. It's a tough job.
Yeah.
Thank you for saying that. You have anything to add,
Yeah,
Terry?
no,
Does
I'm
it
good. It's been great talking to you, Alan.
really
So,
has been. Thank you for sharing your expertise, and please feel free to reach out if you ever want to share any more information. This has been wonderful.
Okay, well, yeah, got me looking back at some of this study, and maybe I'll try to just summarize it up a bit. That would be it'd be good along with all the other stuff that I
yeah.
need to.
Well, if you have free time and you wanted to write an extension letter or article on it, I appreciate everything you've done for us over your 52 years and also taking time out of today to share your expertise with our grower base that's listening. Once it's done, I'll send you the link so you can listen to it if you're interested. But appreciate it again. Thank you for coming out. If you have any further questions, if you're a listener and you want more information, please feel free to email me and I will make sure to direct them to Dr. Lakso to see if he can get some of that data and bring it back to us. And if you wanted any other topics that you're interested in listening to, also please reach out and we can find the expertise behind them to bring to our program. Thank you so very much for joining us today and look forward to a new year. Bye, everyone.
Bye-bye. Thank you.